Hello student, myself Dr. Tanushree Chatterjee from Raipur Institute of Technology. Welcome you all in the SWAYAM course of genetic engineering and its recent advancement. Today I will tell about the DNA fingerprinting. First I will tell about what is DNA fingerprinting, its history, then introduction, what is the principle, choice of cleavage agent, choice of label, methods, cleavage agent, advanced application of it, what is the application of DNA fingerprinting and what are the advantages and disadvantages and recent advancement of the DNA fingerprinting. So, in 1978, David J. Gallus and Albert Smits developed a DNA footprinting technique to study the binding specificity of the lac repressor protein. It was originally a refinement of the Maxam Gilbert chemical sequencing technique. This is the Maxam Gilbert sequencing reaction, cleaving the same tagged segment of DNA at different points yield tagged fragments of different sites. The fragment may then be separated by gel electrophoresis. The various processes in transcription regulation have been studied extensively to determine the sequence specificity of DNA binding proteins. So, the DNA footprinting, which can be defined as a method of assessing the selectivity of DNA sequence to a specific binding ligand. This method was used to find the sequence specificity of various ligands such as actinomycin, echinomycin and related compounds, distamycin and other minor groups binders, metharamycin and many other ligands. DNA footprinting can be used for assessing transcription factors which binds to promoter, silencer or enhancer region of gene to regulate its expression. The first footprinting methodology was developed in the year of 1978. The first method has its application in studying the interaction between DNA binding proteins and their target sites. This method was used to find the sequence specificity of various ligands such as actinomycin, echinomycin and related compounds, distamycin and other minor group binders, methamycin and other ligands. This method is the go-to technique for assessing the sequence specificity of novel ligands. The method is applied to various type of ligands including polyamide, triplex forming oligonucleotides and minor groove binding ligands. Now, what is the principle of the footprinting? DNA footprinting. Footprinting can be simplified as a protection assay of inhibition of cleavage by ligand binding at the binding sites of a DNA fragment. The assay limits the action of digestion by cleavage agent such as DNA is 1 or hydroxyl radicals. A double stranded DNA is labeled at one end to each strand. Then this double stranded DNA is cleaved by a chemical or enzymatic cleavage agent. A single hit kinetics is adapted in the cleavage stage so that on the average each DNA molecule is only cleaved once. The cleaved products are resolved using a denaturing polyacrylamide gel. Since the ligands are selective to a certain sequence, they will protect these regions for cleavage. These products will be missing from the reaction and will be found as a gap that is footprinting in the gel. The exact location of the binding sites for a ligand is determined by these gaps, and they are found by running a control and ligand treated digestion alongside suitable markers. When the concentration of ligands varies over a range in footprinting reaction, its binding affinity can be estimated. In this technique, nucleases like DNase 1 is used, which will degrade DNA molecule. Nucleases cannot degrade DNA if it is bounded by protein, thus that region is protected. That protected DNA region is called DNA footprinting. Now, choice of cleavage agent. The characteristics of an ideal cleavage agent is it should be generate an even ladder of bands in ligand free environment, that is quick, cheap, easy to use, should provide high resolution information about the ligand's preferred binding site. The length of the substrate used for the footprinting are usually 50 to 200 base pairs long. The limiting factor for the length of substrate is the resolution of the polyacrylamide gels. The essential characteristics of substrates fragments should contain the preferred binding sites for the ligand under investigation. When the selectivity of ligands for only 2 consecutive base pairs are easily used for this technique. When ligand selectively increases, there is a chance of the preferred sequence is not included in the fragment. Labeling radioactively is still the preferred method of choice. Fluorescence labeling also can be used. A fluorescent label is employed in case of automated DNA sequencing. The limitation of fluorescence labels is the sensitivity of the label is not applicable in case of where large duplex system is in phantomol. Therefore, radioactivity becomes the most used method of labeling. The fragment can be labeled at either the 3' or 5' end. The source of the substrate and the location of the binding sites relative to the end of the fragments becomes the determinant factor of labeling. When synthetic DNA oligonucleotides are used as footprinting substrates, labeling is carried out at the 5' end. It is done using 32p ATP and polynucleotide kinase. First, the complementary oligonucleotides are designed to produce single stranded overhangs. These overhangs have the property to be labeled at the 3' end by filling with Klinov fragment or reverse transcriptase. The process of direct labeling of restriction fragment at the 5' end is more laborious. This is due to the reason they processes a 5' phosphatase, which hinders the addition of 32p, and therefore, it has to be removed before proceeding to radiolabel. In case of fragment produced by these 2 restriction enzymes, then the enzyme should be added sequentially. After the kinase reaction, only one strand is labeled in the target duplex 5' end. Labeling can be efficiently done by PCR using 5' end primers. 3' end labeling is employed in case of restriction fragments. It is done by filling with 32p DATP or 32p DCTP using a suitable polymerase. Now, in substrate preparation, 5 prime labeling of synthetic oligonucleotides, there are different steps. In first step, in this step both strands of the synthetic duplex are synthesized. One of them is labeled and they are then annealed. One strand is first radio labeled at the 5 prime end using G32P ATP and polynucleotide kinase, that is PNK. In second step, the kinase reaction is usually very efficient and equimolar quantities of ATP and the oligonucleotide can be used, that is 1 ml of 3 millimolar oligonucleotide, 1 ml G32P ATP, 2 ml PNK buffer, 1 ml PNK, that is 10 units, and 15 ml water. In third step, the reaction is complete after incubating and 37 degree centigrade for 30 to 16 minutes. In fourth step, the labeled oligonucleotide can then be separated from any uni-incorporated label by gel filtration. In fifth step, gel purify the labeled oligonucleotide by adding 10 ml DNase 1 stop solution, that is 10 millimolar EDTA, 1 millimolar NaOH, 0.1% bromophenol blue, 80% formamide to the kinase reaction, boiling the reaction for 3 minutes before cooling on ice. In next step, that is the sixth step, load the entire labeled nucleotide onto a denaturing polyacrylamide gel, that is 40 centimeter long and 0.3 mm thick, run at 1500 volt for about 2 hours. Then after electrophoresis, the glass plates are separated and the radioactive oligonucleotide is located by exposure to a X-ray film for about 1 minute. In next step, excise the band containing the radio-labeled DNA from the gel and the radioactive oligonucleotide is eluted by covering with 300 ml, 10 ml 10 millimolar trisaccharin, pH is 7.5 containing 10 millimolar EDTA and gently agitating overnight at room temperature. In next step, the eluted DNA can then be concentrated by ethanol precipitation and redissolved in a suitable volume of TE buffer. Then duplex DNA is generated by adding an excess of the unlabeled complementary strand hearing to 100 degree centigrade and cooling slowly to room temperature. In this step, the annealed duplex can then be further purified in a similar fashion using a non-denaturing polyacrylamide gel. Now 3 prime labeling. Cloning the footprinting template, 3 prime end labeled fragments are prepared by feeling in the sticky ends of appropriate restriction fragment with a 32P DATP. The 3 prime labeling steps are: in case of synthetic DNA fragments, cloning them into the polylinker site of standard vectors is the easiest approach. Second step, cloning of short oligonucleotide duplexes is done by annealing them with complementary synthetic oligonucleotides. In third step, PCR is preferred method for longer fragments. In the PCR method, full length synthetic strand and appropriate primers are used to generate the full duplex. In fourth step, remove the thermostable polymerase from the PCR reaction. Next step, the amplified DNA is cut with BAM HL. Purification This PCR product is done on 2% agarose gel. In 7th step, this is then ligated into BAM-HI, cut PUC-80 and transformed into E. coli Tg2, and white colonies are selected from agar plates containing ampicillin, X-gal and IPTG. In next step, the sequence of the clones must be confirmed by sequencing. Now, summary of the labeling: DNA templates are labeled at the 3' or 5' ended depending on the location of the binding site. Available labels are radioactive and fluorescent. Since this method was originally developed from Maxim Gilbert chemical sequencing technology, radioactively has traditionally been used to label DNA fragments for footprinting analysis. Radioactive labeling is very sensitive and is optimal for visualizing small amount of DNA. Fluorescence is a desirable advancement due to the hazards of using radiochemicals. However, it has been more difficult to optimize because it is not always sensitive enough to detect the low concentration of the target DNA strand used in DNA footprinting experiments. Electrophoretic sequencing gels or capillary electrophoresis have been successful in analyzing footprinting of fluorescent tagged fragment. Various cutting agents can be selected. Desirable agents are sequence independent, easy to use and easy to control. Unfortunately, no drug is available that meets all these criteria. So, the appropriate drug must be selected according to the DNA sequence and ligand of interest. The cleaving agents are DNase 1, that is large protein that function as a double stranded endonuclease, bind is the minor groove of DNA and cleaves the phosphodigester backbone. It is a good cutting agent for footprints because its size makes easy to be physically hindered. Therefore, DNase action is likely to be blocked by binding proteins on the DNA sequence. Additionally, the DNase 1 enzyme can be easily controlled by aiding EDTA to stop the reaction. The use of DNase 1 has some limitations. This enzyme does not randomly cut DNA. Its active is influenced by local DNA structure and sequence, forming a heterogeneous ladder. This can limit the accuracy of predicting protein binding sites or DNA molecules. Here you can see the general strategies of DNA footprinting assays. Hydroxyl radicals are generated from the Fenton reaction, in which iron is reduced with H2O2 to form free hydroxyl molecules. These hydroxyl molecules react with the DNA backbone and cleavage occurs. Due to their small size, the resolution of the resulting DNA footprints is high. Ultraviolet radiation can be used to excite nucleic acids, causing a photoreaction that results in damage to the bases within the DNA strand. Photoreactions include single strand breaks, interaction between or within DNA strands, reactions with solvents and cross linking with proteins. The advantage of using UVs is that they react very quickly. So, you can capture moment to moment interaction. In addition, UV can penetrate cell membrane, making it applicable in vitro experiments. The disadvantage is that the gels are difficult to interpret as the bound proteins do not protect the DNA. They simply alter the light responses of the surrounding. Advanced applications are in vivo footprinting, quantitative footprinting and detection by capillary electrophoresis. In vivo footprinting is a technique used to analyze the protein DNA interactions that are occurring in a cell at a given time point. DNase 1 can be used as a cleavage reagent if the cellular membrane has been permeabilized. However, the most common cleavage agent used in UV irradiation. Once the DNA has been cleaved or damaged by UV, the cells can be lysed and DNA purified for analysis of a region of interest. Ligation mediated PCR is an alternative method to footprint in vivo. Once a cleavage agent has been used on a genomic DNA resulting in single strand breaks and the DNA is isolated, a linker is added onto the break points. A region of interest is amplified between the linker and a gene specific primer, and when run on polyacrylamide gel will have a footprint where a protein was bound. In vivo footprinting combined with immunoprecipitation can be used to assess protein specificity at many locations throughout the genome. The DNA bound to a protein of interest can be immunoprecipitated with an antibody to the protein, and then specific region binding can be assessed using the DNA footprinting technique. Now, quantitative footprinting. What is quantitative footprinting? The DNA footprinting technique can be modified to assess the binding strength of protein to a region of DNA using varying concentration of the protein for the footprinting experiment. The appearance of the footprint can be observed as the concentrations increase and the protein's binding affinity can then be estimated. Detection by capillary electrophoresis. How to detect by capillary electrophoresis? To adapt the footprinting technique to updated detection method, the label DNA fragments are detected by a capillary electrophoresis device instead of being run on a polyacrylamide gel. In the DNA fragment to be analyzed is produced by polymerase chain reaction. It is straightforward to couple a fluorescent molecule such as carboxyl fluorescein FAM to the primers. This way the fragments produced by DNase 1 digestion will contain FAM and will be detectable by the capillary electrophoresis machine. Typically, carboxytetramyl romidine labeled size standard are also added to the mixture of fragment to be analyzed. Binding site of transcription factors have been successfully identified this way. Genome wide assays in next generation sequencing has enabled a genome wide approach to identify DNA footprinting. Open chromatin assays such as DNase sequencing, and they have proven to provide a robust regularly landscape for many cell types. These assays require some downstream bioinformatics analysis in order to provide genome wide DNA footprintings. The computational tools proposed can be categorized in two classes: segmentation based and site centering approaches. The segmentation based methods are based on the application of Hiten-Markov models or sliding withdrawal method to segment the genome into open or closed chromatin region. Example of such methods are HINT, Boyle method and Neff method. In site centric method, on other hand, the find footprints given the open chromatin profile around motif predicted binding site, that is regulatory regions predicted using DNA protein sequence information, that is encoding in structures, for example, centipede and cellular Partioda method. Footprinting with 1,10 phenatryline copper, 1 to 10 phenatryline copper is a chemical nuclease that cleaves the phosphodiester backbone of DNA and generates a nick at the DNA having 5 prime and 3 prime phosphorylated termini, releasing the free base. The small size of the chemical assures a very good resolution, although it is able to cut at all positions. Regardless of the base present, the intensity of cutting does depend on the local sequence TAT, triplets being most preferred because OPCU binds the DNA minor group. It will reveal interaction of the protein with the minor group. A major advantage of this reagent is that protein DNA complexes can be separated in a non-denaturing gel and afterward the gel can be immersed in a solution containing copper, so that the DNA is exposed to the nuclease within the gel. This is useful for labile protein DNA complexes. Now, potassium permanganate footprinting. Some proteins melt the double stranded DNA when binding to it, generating short regions in which the two strands are unpaired. A classical example is that of the bacterial RNA polymerase when forming an open complex at a promoter. Potassium permanganate reacts much more rapidly with single stranded DNA than with double stranded DNA, being rather selective for thymidines. This makes potassium permanganate useful at probing the melted region, provided that there are thymidines in it. The footprinting reaction does not cleave the DNA, but just modifies the unpaired thymidines. Therefore, after stopping the footprinting reaction, the DNA is precipitated and cleaved at the modified position by treatment with 1 molar piperidine 30 minutes at 90 degree centigrade. The cleaved fragments are then analyzed by electrophoresis in urea polyacrylamide gel. The melted region appears as a number of hypersensitive thymidines in 1 or 2 DNA strands. Next one is dimethyl sulfate footprinting. Dimethyl sulfate methylase guanine residues at the N7 position and with a reduced efficiency. Adenine reduces at the N3 position. DMS can be used for methylation protection or methylation interference assays. DMS can be used for methylation protection or methylation interface assays. In protection assays, the protein DNA complex is formed before addition of DMS and the footprint indicates which guanine or adenine residues are protected from DMS attack by the bound protein. Since DMS readily penetrates intact cells, methylation protection can be performed both the vivo and in vivo and in vitro. When performing the footprinting in vitro, a DNA fragment label at just one end is used, adding DMS at the concentration that allows methyling. On average, only one position per DNA molecule, since DMS methylates the DNA but does not cut it. After the footprinting reaction, the DNA is cut at the methylated position by treatment with piperidine as described for KMNO4 footprinting. Now, what are the application of DNA footprinting? The classic application, the present technique has to study protein DNA interaction. The means that the site where a particular factor binds can also be assessed and determined. The transcriptional regulation and gene expression processes can be studied. The location of various transcriptional factors like promoters, enhancer and regulatory can be located effectively. It can also be used to identify the function piece of sequence from the genome. Recent advancement also show that hormone response elements and their binding to specific sequences can be investigated. It also facilitates drug DNA or ligand DNA interaction studies too. Now, what are the advantages? Advantages of footprinting, DNA footprinting. The present technique is powerful enough to differentiate many fragments. It can exactly locate the binding site of the particular ligand. The present technique is a comparatively cheap, and some disadvantages are also there. The present technique is a conventional, time consuming and laborious. It is less accurate and non reproducible. Binding resolution is very low, no so robust. Radioactive use is hazardous for health. Then what are the recent advancement of DNA footprinting? Although DNA footprinting has lucrative application in molecular genetics, it comes with serious drawbacks. Recent automation explained by Yanadisen and Shell 2000 suggests that the use of fluorochrome ready to use DNA extraction and purification kit and capillary electrophoresis increases binding resolution accuracy and safety, also is robust and rapid. Auto radiography like TDS step is a highly sensitive and robust technique that can differentiate a single base difference accurately. Some researchers also use gel shift assay to which illustrate differences in the mobility of protein linked and non-linked DNA under electrical current. These techniques are insufficient in providing more data. So, in conclusion we can say DNA footprinting studies epigenetics and gene expression, which is indeed a important factor for producing variabilities. Chromatin interaction with protein ligands for hormones alter the structural stability of DNA as well as its functionality greatly. The euchromatin to heterochromatin and vice versa conversions are regulated by protein or transcriptional factor that can be studied through DNA footprinting. Thank you.